This is our 7th lecture of the semester, and this is part 2 of discussing protein structure, and that's material from chapter 4. On the title slide, this is a figure from a different source and a review of all the different layers, the hierarchy of protein structure. Remember, we have primary structure, which is the sequence of amino acids. Secondary structure are these local structures, primarily due to the hydrogen bonding between the backbone, carbonyl and amino groups in the peptide backbone. Okay, and those are the beta sheets or beta strands and the alpha helix primarily. Okay. Today we're going to be talking about third and fourth order structures. Okay, so the tertiary structure of a protein, that's the overall folding pattern of a protein, and this is really more due to side chain interactions. Okay, secondary structures we typically think of as due to backbone interactions. Tertiary structures are due to side chain interactions. Okay. And then quaternary structure is when you have two or more polypeptide chains coming together to form a complex. And just a reminder, this is section 3 and section 4 of chapter 4. Tertiary structure refers to an overall spatial arrangement in 3 dimensions of the atoms in a protein. Okay. And a tertiary structure is going to be stabilized by numerous weak interactions. And primarily we're thinking of these as due to the side chains of the amino acids. Okay. Hydrophobic interactions play a huge role. Other polar interactions as well. So if you have hydrophobic amino acids, they tend to get buried in the 3-dimensional structure. Polar amino acids will tend to either be on the outside of the protein that is exposed to an aqueous environment, or if they are buried, polar residues will be interacting with another polar residue by a hydrogen bond or something like that. Also, the tertiary structures of proteins can also be stabilized by disulfide bonds. And that's a very specific interaction between 2 cysteine amino acid side chains. The interacting amino acids in a tertiary structure are not necessarily next to each other in the primary sequence. So remember that amino acids that are very far away in the primary sequence, really, they can be on opposite ends of a protein sequence. When the protein folds into a 3-dimensional shape, those 2 amino acids could be right next to each other and interact. There are 2 major classes for proteins based on tertiary structures. There are fibrous proteins and globular proteins. We tend to think of fibrous proteins as being more lipid soluble, and globular proteins as being more water soluble. So we'll first look at fibrous proteins. Okay. There are 3 types of fibrous proteins that we're going to talk about in the class, and those are listed here in table 4.3. Okay. They're actually listed on the right side of this table. So alpha keratin, the silk, fibronin, and collagen are the 3 types of fibrous structures that we're going to look at. Okay. What primary structure they're made of is listed here on the left side of the table. Okay. So alpha helix, a cross-link alpha helix, that's the main structure that alpha keratin uses. A beta conformation is silk fibronin, and what's known as the triple helix or collagen triple helix is the structure collagen uses, and their characteristics are defined here. All three of these are really considered pretty strong structures. So their three-dimensional structure and fourth-dimensional structure, if you think about the quaternary structure of these, it really leads to a very strong material in all cases. Some are more rigid, like the alpha keratin, and others like silk fibronin are softer and more flexible, but they're all very, very strong. So if we look at a cross-section of hair, this is what we would see, sort of a cartoon type picture of this cross-section of hair, right? And you would notice you have these cells, and then if you zoom in a little bit closer, you notice that there's actually filaments in here, and the filaments have, many orders of structure. And what makes up ultimately these filaments are alpha helix, two alpha helices that are wrapped around each other. So it's a two-chain coiled coil is what it's referred to as. We're going to see this coiled coil some more. So if we look at this, this is alpha keratin, that would be the protein in hair and nails, even on the outside of your skin. Okay. I want to mention too that if you ever see a molecule with the name, a name that has IN at the end of it, that typically means you're talking about a protein. So the alpha keratin are made up of two alpha helix chain, and each one of them is a coil. An alpha helix is a coil, but they're wrapped around each other in another coil. So we call this a coiled coil. And you'll see it depicted in a cartoon version like this, where you have these bigger ends at the bigger globular piece, sort of at the end of the filament, at the end of a coiled coil. And these stack end to end with each other, these coiled coils, and that's what makes up. If you have two chains of these, that makes up a protofilament, and the cross section of that would be about 20 to 30 angstroms. And if you remember, the width of an alpha helice is typically something around 10 angstroms. So here, wrapping these alpha helices around each other, you can see that it's about 20 to 30 angstroms wide. Okay, and then the protofibril is just more of these protofilaments, two of the protofilaments wrapped around each other. Okay, and much like if you got a steel cable, a braided steel cable, that's what this alpha keratin is doing. You have individual alpha helices that are wrapped around each other, sort of braided, and then you have those braids braided around each other in a protofilament, and then protofilaments wrapped around each other in a protofibril, and so forth. And you can imagine that this makes a very strong structure. The book talks a little about the chemistry behind a permanent. When you have a perm in your hair, this is the chemistry behind that. And we mentioned that in alpha keratin you have alpha helices wrapped in a coil, but then there's also disulfide cross-linking between the alpha helices to make that stronger. Okay, what you do in a perm is you first have a chemical that reduces the disulfide bonds. So as we start here on the left side, this is what we would refer to as the oxidized form of a disulfide bond. that's when there is a disulfide bond between two cysteines. So that's its oxidative state. When you reduce that, you're adding hydrogen. Okay, if we think back to general chemistry, reducing something means you're adding electrons, oxidizing means you're taking them away. In organic molecules, the adding of an electron around a bond typically manifests itself by the addition of a hydrogen as well. Okay, so that's one way, and this helps when you go into metabolism. If you try to look and see, is this molecule being oxidized or reduced, oftentimes it helps if you see hydrogens added to the molecule. You can tell that molecule is being reduced. Okay. So when we reduce those disulfide bonds, that breaks the disulfide bond. Okay. So now these strands are the cysteine residues aren't linked together. Okay, you do that, you curl the hair, so now the hair takes a different shape. When it does that, the cysteine residues aren't lined up exactly like they were in the straight hair. Then you oxidize them, right? And oxidizing them makes the formation of these disulfides, but now they're at different positions, and being at different positions, that makes the hair hold a different shape, in this case a curl. Okay, moving on to collagen. Okay. Collagen is important in connective tissue, so things like tendons and cartilage. Okay. The collagen chain is a long helix, but it's a little bit different. It's a left-handed helix. So remember, the alpha helix is a right-handed helix. In collagen, each chain is a left-handed helix, and they're rich in glycine and proline amino acids. And remember, glycine and proline were amino acids that weren't really good for the formation of alpha helices. Okay. 3 collagen chains interconnect to form a right-handed superhelical triple helix. Okay, so this is a little bit confusing, right? Each collagen chain is a left-handed helix, but they wrap into each other in a right-handed fashion. Okay, so you have a left-handed helices that are wrapping together and twisting right-handedly. Okay, so this is a very sort of complicated idea or complicated structure. Okay. The triple helix is very strong, higher tensile strength than steel of an equal cross-section. Okay. And many of these triple helices are assembled together into a collagen fibril. Okay. This is the picture of that. So these individual collagen chains are colored here in different colors, so white, blue, and purple. Okay. The left-handed helice, and you can look at this and make sure, right? Do the hand rule. Your thumb goes in the direction that the helix is going, and then you curl your fingers. You can see that is left-handed. Okay. But those curl together in a right-handed fashion. Glycine and proline rich sequence. There's also another amino acid, one of the uncommon amino acids, the 4-hydroxyproline. That is important in collagen fibrils. So 4-hydroxyproline in collagen forces a proline ring into a favorable pucker. Okay, that offers more hydrogen bondings between the 3 strands. Okay. This is done post-translationally, and it's catalyzed by an enzyme prolyl hydroxylase. That enzyme requires a couple things, or three things actually: alpha-ketoglutarate, molecular oxygen, and ascorbate. Ascorbate is the another term for vitamin C, another name for vitamin C. And why does 4-hydroxyproline, why does this change the structure? Well, if we look at the structure of a proline, that the gamma carbon in the endo position is shown here, in the exo position is shown here, and that adding the hydroxy group really forces it into that exo position, which is beneficial for the hydrogen bondings between the three chains. Okay. Wouldn't expect you to be able to draw this. Showing you a picture of this, I think hopefully you would be able to associate this with the 4-hydroxyproline, and that being important in the collagen triple helix. Okay, this is in the text. This is in a sort of a what they call a box, a green box in the text, and going through the details of this reaction. It's a little bit confusing though, as to just by the reactions that the book shows why ascorbate is so important in this. If you just look at these, right, because this reaction, when you have proline, alpha-ketoglutarate, oxygen, and you get 4-hydroxyproline and succinate, like why the book doesn't do a good job of explaining why ascorbate is necessary. Okay, so I wouldn't expect you to recreate this on an exam. If I asked you a question about this, knowing that ascorbate is important in collagen, I think would be what would be important to remember. Okay, and then the book also talks about vitamin C deficiency in scurvy, and that, you know, one of the reasons why the disease is so bad is that, you know, your connective tissue, your collagen, basically gets weaker and degrades because you don't have that ascorbate, which is ultimately needed to produce the 4-hydroxyproline post-translational modification in collagen that makes it so strong. Okay. So collagen superstructures are formed by cross-linking these triple helices together to form what are known as collagen fibrils. And here's an electron micrograph of collagen fibrils. And what's going on in these is you have these triple helices. So you have 3 collagen chains interwound in the triple helix. And you have lots of these, but then they're being cross-linked together. And that's forming a very strong material. Okay, and those cross-links, unlike in alpha-keratin cross-links are disulfide bonds. The cross-links in collagen are between lysine or hydroxylysine and histidine amino acid residues. Okay. So that's a difference as well, right? So if we think of, these are both coiled structures, but, you know, if we think about the differences, alpha-keratin is alpha helical, right-handed helices at the main starting point of the structure. Collagen would be a left-handed helix. Okay. That's one major difference. Another difference would be the cross-linking. In alpha-keratin, you have disulfide cross-links. In collagen, it's lysine or hydroxylysine and histidine. Okay, the last fibrous protein that we're going to discuss is silk fibronin. And then here we see that IN again in fibronin. That tells you it's a protein. Okay, fibronin is the main protein in silk from moths and spiders. And it, unlike the alpha keratin or collagen, it's the structure, the primary structure that fibronin is using is a beta sheet. Okay, small side chains like alanine and glycine are the main amino acids in fibronin. And they allow these beta sheets to be closely packed together. The structure is stabilized by both hydrogen bonding within sheets and then also dispersion interactions between sheets that are stacked very closely on top of one another. So here we see a depiction of fibronin, and in each color is a different layer, a different beta sheet that are stacked on top of each other. So this, and they're I should mention that too, they're anti-parallel beta sheets. So the strands are going in alternating directions. If we look at the top layer, so this is the top layer in red, if we look at the ball and stick model of this, right, we have our alanine side chain shown here in black. Okay, you're alternating alanine and glycine in these amino acid chains. Okay, and then within a sheet, okay, if we're looking at the same molecule position within a sheet, okay, we have an alanine side chain facing down, alanine side chain facing up. Okay, alanine side chain facing down, alanine side chain facing up, and so forth in that sheet. That stacks with the next sheet, okay, and then the alanines, okay, are slightly offset so that the side chains can move a little bit further or closer to the main chain in each case. So it's really like an interlocking, almost like a zipper, really. It just fits really tightly together. And that's where the dispersion forces come into play. All right, so this silk, in the case of a spider, right, spiders use their silk to make their webs, their egg sacs, and if they catch something in their web, like a bug, they wrap that bug up in the silk. It's an extremely strong material, okay? Just like we saw in collagen and alpha keratin, it's stronger than steel when you're looking at per cross-sectional area of this. What's different about the fibronin in the spider silk is that this material is very stretchable, very flexible. Okay, it can stretch a lot before it actually breaks. And spider silk isn't just the fibronin protein, it's actually a composite material. It has a crystalline part, which is very rich in the fibronin protein, and then there's other rubber-like stretchy parts to it. So spider silk is really an example of a composite material. Okay, so that's fibrous proteins. What we're going to discuss now are globular proteins, the other main type of three-dimensional overall three-dimensional structure of proteins can be fibrous or globular. Okay. We tend to think of globular proteins as being the water-soluble proteins. Okay. If we took a protein sequence, and we force that all into the beta conformation. And remember, beta conformation is pretty extended. Okay, so in a beta conformation for a given length of a sequence, if it was all beta conformation, this would be a very long structure. and the dimensions are shown here. Okay, I forget how many amino acids this is, but however many this is, it would be about 2,000 angstroms long and only 5 angstroms in diameter. Okay. If you take that same sequence and force it into an alpha helical conformation, it shortens it and it makes it a little bit wider. So this is 900 by 11 angstroms. Now, this same structure that it's in its native fold, a globular form, okay, and this is myoglobin. Its dimensions are 100 by 60 angstroms. So you can see that you're taking this string and you're compacting it into this area. So globular proteins are very, very compact structures. We tend to think of globular proteins as being made up of certain what we call motifs or folds. Okay. And those two words are a little bit used interchangeably, but what we would think of a motif as being being a little bit smaller structure, and a fold as being maybe the overall fold, the overall three-dimensional shape of a protein. So globular proteins have a specific arrangement of usually several secondary structure elements, okay, either alpha helix, beta sheet, or typically a combination of both. Okay. And certain motifs can be found as being reoccurring structures in lots of different proteins. Okay. And in fact, lots of proteins with different sequences can have similar folding motifs. Okay. Globular proteins are composed of different motifs combining together to give you an overall shape or an overall fold of the protein. Okay, so some examples of motifs are shown here. This is known as a beta alpha beta loop. So the sequence, if we follow the sequence in order, it's making a loop. Okay? And it starts with a beta strand segment going into an alpha helix and then into another beta strand. So beta alpha beta loop. Okay, this guy is known as a beta barrel. You also hear this referred to or see this referred to as a TIM barrel. And that stands, the TIM, T-I-M, it stands for triose phosphate isomerase barrel, okay, which is the name of the protein that it was first found in. Okay. So it's really some beta strands that are twisted around each other to form a barrel. Okay. So those would be two motifs. And then motifs, there are many different motifs. These are just a couple examples. Okay, the book talks about a database that shows really tries to catalog all the different motifs that have been found. Okay, some motifs are found in one protein. Other motifs are found in thousands of proteins. Okay. But in some proteins, you get repeated motifs. So in a case of the beta alpha beta loop, that can be repeated several times within a protein and give a final fold. Okay, so this would be the final fold. And you see this, and this is known as a alpha beta barrel. Okay, so an alpha beta barrel is a fold that's made up of different beta alpha beta motifs. Okay, and the alpha beta barrel, you see that typically on the inside of that barrel is a binding site for a certain ligand, a certain small molecule that will bind in there. Well, there's also proteins that are disordered, and it's not that they're they're disordered for a certain time and then adopt a certain structure, they're always disordered, or at least parts of them are always disordered. And when you look at a crystal structure of the protein or an NMR structure of the protein, the crystal structure, that disordered region doesn't show up. Or in an NMR structure, it's very mobile, okay? And we refer to those as being intrinsically disordered. And you can also think of that as lacking definable structure. It still has it's not, I need to be careful here. It's not that it's, necessarily wildly moving all over the place. It's just that it's flexible and it doesn't have a specific secondary structure or more motif that we can use to characterize it. Okay. So no definable structure. Amino acids that in higher concentration can force this less defined structure are things like lysine and arginine, glutamate. So, right, those are positively charged side chains. Glutamate's negatively charged, and then proline as well. Disordered regions can be used in a protein to interact with numerous partner proteins. So they can being disordered or not having a definable structure, they can interact with multiple different proteins because they can conform in many different ways. And the book gives this example. And this protein is P53, which you might have heard of. It's mutations in P53 are implicated in many different cancers. Okay. So this is P53, which it's not labeled in the figure. Okay. And on the N-terminal and C-terminal of P53, there are these intrinsically disordered regions. And this is a plot of the Ponder score. Ponder stands for predictor of what is that? Predictor of non-definable I forget. Jeez. Yeah, it's not coming to me, but it's a predictor of when something's disordered. Predictor of something disordered regions. Okay, sorry about blanking on that, but you can see that it's when that score is higher, that means that region is more likely to be a disordered region. Okay. And the scores lower down here, the structures in those regions are very defined. Okay. And P53 uses these regions to interact with different partner proteins. So you can see when it interacts with a partner protein, here in orange at the top, when it's interacting with cyclin A, it's forming a very ordered structure in that with that interaction. Okay, so it's intrinsically disordered in the native state, but when it's interacting with a partner, it becomes ordered, and it can adopt different conformations, and they're just shown here in different colors. P53, this partner molecule it's interacting with, cyclin, that name will kind of suggest what it's important for. Cyclin, it's a protein that's involved in cell cycle regulation, which is why if you have problems with P53, the cell cycle can be unregulated, which then can lead to cancer. Quaternary structure, as I mentioned before, is the assembly of individual polypeptides or individual proteins into a larger cluster, a larger functional cluster. And this is an example here. This picture is of hemoglobin. In hemoglobin, you have a pair of alpha beta polypeptides that then make a dimer. So there's 4 main polypeptides in a hemoglobin molecule. 2 of them will be what we refer to as alpha subunits, and 2 of them would be beta. So there's 2 alphas and 2 betas in the 4 polypeptide chains that make up a hemoglobin. And we'll talk a lot more about hemoglobin in chapter 5. Okay. What's important to note now is just the idea of the quaternary structure, how you have a polypeptide, what we would think of as a 3-dimensional functional protein. They can combine together to make bigger complexes. And you can have an example of, the same exact polypeptide interacting with more copies of itself. Or you can have more complicated quaternary structures that have many different polypeptide chains coming together to form a complex. How do we get these pictures of proteins? And that really becomes, it's really important in structure, what we call structural biology. Seeing a structure of a biomolecule and then being able to determine its function. And this all stems from being able to get things like protein structures. The main way we get a protein structure, the primary way, is by something known as X-ray crystallography. Okay, and the book kind of details what steps we need to do this. Just briefly, they're listed here. Okay, first, you need to purify a protein. Most proteins, I think, can be purified. Some are harder to do than others. Typically, purifying a protein involves doing it with recombinant DNA, and that is taking the gene for the protein sequence that you want to look at and putting that gene into some other organism. Most commonly, it would be a bacteria cell, and then you can have the bacteria cell make a lot of that protein. Then it's much easier to purify. You don't really see proteins being purified from their native source, like a human protein. You don't see people taking a lot of human tissue to try to get that protein out, really. Since in a sense, every protein the genetic sequence is known for, you usually do it recombinantly. Okay. And that's the first step. The second step is you actually have to get a crystal of the protein, and that can be very difficult for some proteins, and there's lots of tricks, and that can be a whole course into itself, really. And it's almost more of an art form as well. Okay. Assuming that you get a crystal, you can take the crystal and you can put it in your X-ray crystallography instrument. So you hit it with a beam of X-rays, you get a diffraction pattern, so you collect that diffraction data, and then from that data, you calculate electron density. Hey, this is done, with a computer program, and then you fit known amino acid residues into that electron density, and then doing that, you build up a structure from your data. The pros to crystallography is that there are no size limits to the protein or protein complex you can look at. Okay? And this is a very well-established technique. It's been used I talked a little bit about last time Linus Pauling using this, and that was to get the structure of the alpha helix, and that was in the 1930s, I believe. So this has been used for, going on, at least in terms of using crystallography on biological molecules. It's pretty close to being used for 100 years now. Okay. The cons of it is that it's difficult for membrane proteins, and we'll see when we go on to chapter 10, there's actually a lot of proteins that are membrane bound, that are in membranes. In crystallography, it's just very hard to do with membrane proteins. It's hard to crystallize membrane proteins. Okay. You can't resolve hydrogens as well in crystallography. They're too small to be resolved by the X-rays. Okay, so that's another con. And I think the overall con as well for crystallography is that it needs crystals. Some proteins are just very, very difficult to crystallize. And sometimes you have to use certain conditions to get the crystals that aren't that put strain on the protein that forces them into a non-native fold in the crystal. And here's the figure from the book that just shows you kind of the steps in this. You get a diffraction pattern that's used. A computer program will give you electron density based on that diffraction pattern, and then you can fit in that diffraction pattern your certain amino acids, or in this case it looks like the heme group, and then the iron in the center there of this heme. Okay. The second major way protein structures are determined is by NMR, and NMR stands for nuclear magnetic resonance. Okay. The steps needed here, similar at the start, right? You have to purify your protein, okay? And then your protein, in this case, it needs to be soluble. There are some techniques where the Nmr structure of membrane-bound proteins are done, but it's trickier. You tend to think of your protein has to be in solution to do NMR on it. Okay, you collect NMR data and assign NMR signals, and then you calculate the structure. Okay, I should mention too that NMR, nuclear magnetic resonance, is really a very similar technique as MRI, magnetic resonance imagery, that you would go and get an MRI on your knee. It's using the same basic science in that you're using nuclear spins to get data. So the pros of NMR: you don't need to crystallize the protein, which is a very big plus. In NMR, you're using the nuclear spin of hydrogen atoms, so that's what you're using here. And you can see many hydrogens in a protein. Okay. It's very difficult for insoluble proteins or membrane-bound proteins this way, unless you can somehow solubilize them. And the other main drawback for NMR is that there is a size limitation. It works best with smaller proteins. I don't remember the exact size cutoff, but there is a size cutoff with NMR. And here's an example of some data that you'd get out of an NMR experiment, and typically they're two-dimensional NMR. And from that data, you can have programs that then assign a structure. They get distances between hydrogens. And there's a couple different NMR programs, or I should say experimental techniques that you would do, like NOSI and TOSI, but those it's really not important for our class. Okay, the NMR structure, what's nice about it when you finally get it is that it gives you an idea of how mobile certain parts of a protein, certain regions are. So this region here, all of these, it gives you sort of a composite structure that you lay on top of each other. So you can see that this region here seems to be pretty structured, pretty stable. In other regions, there's more deviation. Okay, and that kind of tells you there might be some more mobility in those regions. Okay, the third one that I'm including here, just because I've done it, is something called electron paramagnetic resonance, or EPR. And this is a very, very closely related technique to NMR, but instead of nuclear spins, you're using electron spins. Okay. And you'll typically, to do this, you really need unpaired electrons. Okay, and to do that, you add what are known as spin labels. So molecules like this have this nitroxide part to them, right, that has an unpaired electron, and they can be visible in EPR spectroscopy. Okay, and this part will bind to cysteine residues, so you can introduce non-native cysteine residues in a protein and put a spin label on it. And then if you do a couple spin labels, you can actually, there's EPR methods that you can get distances between two spin labels. This way, it's not really to get an entire structure of a protein, like a whole three-dimensional structure, but you can use it to get some information on the mobility of a region of a protein. And so when I was in grad school, that's what I did with this molecule, ETF. Okay, and this is the overall three-dimensional crystal structure of ETF. Okay, it has these two colors in it that shows you that it's made up of two polypeptide chains. Okay, so one was alpha and one was called beta. Okay, those come together to form the fully functional ETF. Okay, so that's an example of a quaternary structure. It also had a cofactor, a flavin molecule. So what we did is we reduced the flavin, and the flavin is used to transport electrons in this protein. So we could reduce it into a one electron reduced state that was able to be visualized in EPR, and then we could add spin labels to different parts and get distances between them, okay, using a technique called DEAR. And that's, kind of not really important for the overall message of the class, but just thought it would be something good to include here, something that I've actually done personally. Okay. The last section of this chapter talks about stability, folding, and really unfolding of a protein. A protein's function depends on its 3D structure. So loss of structural integrity will come with a loss of activity of that protein. And when that happens, we call that denaturation. Okay. So that's when a protein loses some structural integrity or unfolds and then becomes inactive. Okay, things that can cause this. Heat is one thing. Extreme cold can also cause this. Okay. Extremes in pH, so very high pHs or very low pHs. Organic solvents. That's one way. If you're looking in a lab at a tissue sample, a lot of times before you run that sample on an instrument, you want to get rid of the protein in it. What you'll do typically a lot of times is you'll add a bunch of organic solvent like a methanol to it, and that causes the proteins to misfold and to be precipitated, and then you spin them out. Okay. And then the last thing is what they refer to as chaotropic agents. The typical ones used are urea and guanidinium hydrochloride. This is just a fancy way of saying salts, really, because that's what you're doing. These just have, are fairly large salts. But if you added enough sodium chloride or potassium chloride to a protein solution, at a certain point they're going to precipitate out, or at least misfold and unfold and denature. Right. In your cell, the amount of protein, native protein that's around at any particular time is referred to as proteostasis. Okay? And this involves many different factors, many different competing factors. Okay, the synthesis of your protein on a ribosome, assembly of it, and the degradation of it all come into play here to how much native protein you have at any given moment. So if we look at our example, this figure, a protein is synthesized on the ribosome. Okay, that protein comes out. I should say right here. Okay, that protein is coming out, and it can fold into an intermediate. And a lot of proteins can just kind of come into their own native folds by themselves. Okay, which would kind of jump over here. But there are other proteins that need help folding all the way to their native state. Okay, so going from an intermediate as synthesized into the native state requires what are known as chaperones for some proteins. Okay, so then we have our native protein. Okay, this native protein can unfold back into the intermediate. It can unfold into what's known as a misfolded protein, and we sort of think of a folding intermediate in the native state as being in equilibrium. You can go back and forth, but a misfold is one direction. Once the native protein goes into that misfold, it can't just go back to the native protein in a single step. Okay, so when that protein goes into the misfold, then in a cell, typically it can be tagged with something called ubiquitin. Okay, ubiquitin. I always have trouble saying that one too. And then that tags it and targets it for degradation. Okay. There's also in native proteins, some proteins hang around for a very long time. Years. I think there are certain proteins in your eyes that have been around most of your life, but that's not really typical. The half-life of proteins, it varies. Okay, some stick around, several days. Others are more like several hours, but some are targeted directly from their native state to be broken down and digested. Okay. Others will misfold, and then that misfolded protein can be then, remodeled back into an intermediate and back into the native state by chaperones. If the misfolded protein, if it happens in enough concentration, what you'll get is aggregation. Okay, so when misfolded proteins aggregate, you get what's known as amorphous aggregates, which would really just be kind of like a precipitation, oligomers, or amyloid fibrils. Those are all kind of examples of some protein aggregation. Okay, and then that in your body will trigger a response that sequesters those aggregates, removes them, and then digests them into peptide fragments. Okay, so this whole process is the process of proteostasis. If we look at denaturation, Okay, we mentioned there are four ways that proteins can denature. Okay, one is temperature. There was temperature. There was pH. There was salt concentrations. Okay, what was the fourth one? Oh, organic solvents. In an organic solvent, you're not going to run into that situation inside of a cell. Okay, so there are four ways in a lab we would denature proteins. Inside of a cell, there's really only three that you would have to worry about. Okay, but when we do these experiments in a lab, we can get a graph of this. So the percent unfolded of a protein by looking at certain amino acid side chains, the absorbance level of them, okay, with respect to our denaturing conditions. So here's an example of temperature. So if you slowly ramp up the temperature, you'll watch the protein unfold, and I guess it's best to look at the blue trace, right? Until it slowly unfolds until it reaches a point where there's this rapid change, and then it will bend over and fold over and flatten out. Okay, this is an example of ribonuclease A. Okay, and that if you do the derivative of this and find the maximum there, we would say that would be in this case, since it's with temperature, we refer to it as a TM, or a melting temperature. Okay, it's not really melting, but it's unfolding. Okay. This is showing the same thing, but now instead of temperature, we're using guanidinium hydrochloride concentration and doing different guanidinium hydrochloride, titrating that in until we see the same sort of unfolding. Okay. What this is showing you, the importance of this, is that the protein it's not a straight line. Okay, it's not unfolding at a constant rate. It's unfolding slowly until it reaches a certain point, and then bam, it really unfolds quickly. Whether it be a few degrees in temperature, or just a few slight changes in concentration. And what that's telling you is that the folding of a protein is cooperative. Okay, there's a lot of cooperation going on involved in keeping that fold together, and that's why it's so hard to break it. And then at a certain point, when you break that cooperation between, and the way you can think of it is that there's these defined regions or motifs that are folded very strongly, and then they come together and interact with one another. Okay, so once you break the interaction of those regions or motifs, then it becomes much more easier to break and fully unfold the protein. A very important experiment was done with ribonuclease unfolding and then refolding. Okay. Ribonuclease, it's a small protein. It contains 8 cysteines. Okay. The 8 cysteines are all involved in disulfide bonds, so they form 4 disulfide bonds. In the presence of urea and 2-mercaptoethanol, or that's also referred to in the lab as beta-mercaptoethanol, it fully denatures ribonuclease. Okay, so the urea, the urea will break hydrogen bonds, that salt, and denature the protein that way. But the beta-mercaptoethanol, that is used for breaking disulfide bonds. Okay. So you can denature the protein fully. When those are removed by dialysis, so the urea and beta-mercaptoethanol are removed, the protein refolds, and it refolds in the correct pattern and the correct disulfide bonds are reformed. Okay, so you can, when Afnison did this experiment, right, the protein, when it was denatured, was inactive. When he removed the urea and mercaptoethanol, it refolded and it gained its activity back. Okay, and so from this experiment, the conclusion was, the protein sequence alone is what's determining the native conformation. Because, if it was somewhat random, right, or let's say when this ribonuclease was being synthesized, if something else was telling it how to fold as it was being synthesized, that wouldn't be around. When you unfold it and allow it to fold back randomly, you would think that you would get different disulfide pairings, but that's not what was seen. The same exact disulfides were formed, and it was active again. Okay, a very simple experiment, but it led to a Nobel Prize in chemistry for this. And just to give some numbers here, okay, if there were 8 cysteines, okay, that would be something like 105 possible disulfide bonds that could form. Okay, but when they did this experiment, only the 4 correct ones were folding. Okay, so this is the figure depicting this experiment. So this is our native state that's active. Okay, we unfold it and remove the disulfide bonds, so it's disordered. When you remove the urea and mercaptoethanol, it folds back into the native state with the exact same 4 disulfide bonds. Okay, so there's something inherent in this structure, this primary structure, the sequence of this protein, that tells it how to fold. Okay, and proteins, and that's really kind of a very important thing to realize, that the sequence of a protein ultimately tells it or guides it into how it's going to fold. So I think really the goal and the missing step from being able to say, here's the DNA sequence, the amino acid sequence comes from that, and then that amino acid sequence, now where's you just going from an amino acid sequence, being able to go from an amino acid sequence into what a 3-dimensional shape and function of that protein is, that's kind of the missing step. Okay, so how can we predict from amino acid sequence to protein sequence and function? And that's really becomes the hard part. People have tried to research this, how proteins fold, and one of the, I guess, consensus right now is that folding follows a distinct path, okay, but the actual steps involved can be slightly different, depending on protein or depending on conditions. And what you notice when people have studied this, what they've noticed is that there are certain regions that give, pretty stable secondary structures or motifs. And it's thought that they will sort of fold first, and by those folding, then they will direct the rest of the protein around them into folding. Okay, so you sort of hop into these different secondary structures given a certain sequence. Okay, certain parts fold first, they're very stable, and then it directs the protein on how to fold from there. Okay, so there's a direction to it. It's not random. Okay. And that's where we get this idea of Leventhal's paradox. Okay. Proteins fold to the lowest energy, okay, and they do this in microseconds to second time scale. Okay, that's pretty fast when you consider how many amino acids are in a protein and how many degrees of freedom they have. Okay, so just to give some numbers on this, if I can do this. There we go. Okay, so say you have a typical protein is something like 100 amino acids. Okay. Each of those amino acids has about 10 possible conformations if we look at a Ramachandran plot of the phi and psi pairs. So about 10 conformations. Right. Or I shouldn't say that each, okay, so that's for each amino acid has 10 conformations. When we combine them together, okay, that would be 10 conformations to the 100th power. There's 100 amino acids each, so each can have 10 conformations. So the protein, in theory, could have a total of 10 to the 100th power conformations. So that would be the protein conformations possible. Right? Now, if you assume that the protein can sample conformations on the same time scale as bond motion, so something like 10 to the minus 13 seconds it takes for that bond motion. Okay, so if you have 10 to the 10 conformations and you multiply that by, you can do one conformation sampled every 10 to the minus 13 seconds. That still gives you something like 10 to the 77th power years it would take. Okay, so if a protein is just randomly sampling conformations at every amino acid till it finds the lowest energy, it would take 10 to the 77th years to fold correctly. And we know that's not possible, right? Because we see proteins fold in our daily lives. Okay, so this search for the minimum energy structure is not random. Okay, the direction towards that native structure is following a direction of thermodynamically most favorable steps. And that's where this idea of a free energy funnel comes from. Okay, so you're not just randomly trying to get to the native structure, right? So if you start at, let's say here, and you're randomly going to there, you would kind of be bouncing all over the place. But it follows more of this surface, a funnel that drops you down in free energy wise to the native state. And there's different types of funnels. Not important to know the differences between all of these. I think the most common one you see is something like this that has these ridges in it, and those would be, you're following this part, and then you have to go, certain directions to avoid the more unfavorable types of folds in proteins. And so this is probably more realistic, right? Because when we talk about folding intermediate, it might be something up right here that's trapped by this ridge of higher free energy. So it has to find a way to get around that, and maybe a chaperone helps it move around that to the native state. Okay. And that's where chaperones come in. The book talks about two different types. One is the HSP70, and the other is chaperonins, or heat shock protein, HSP60. Okay. The first, HSP70, that HSP stands for heat shock protein. 70 is just the molecular weight of that, and there's a whole class of these HSP70s. They tend to be expressed more when the temperature goes up. That's why they're called heat shocks. If we have our folding intermediate here, we want to get to our native state, but sometimes that's not possible directly, and chaperones help. In the case of heat shock protein 70, it'll be in an open conformation with ATP bound, okay, and that's where it can bind our folding intermediate. It usually will bind to hydrophobic parts of a protein, and you can have lots of these HSP70s on a protein that's being synthesized. So typically the protein will be synthesized on a ribosome, and these HSP70s will bind to the hydrophobic regions of that protein and help them fold. Okay, so it binds there. Okay, that can go into then once it binds there, a protein HSP40 can bind, and that hydrolyzes ATP. Okay, knocks off the phosphate from that. Okay, that closes the HSP70, makes it high affinity, so traps the folding protein. That folding protein can then, when it's trapped, can help be directed to the proper fold, and then a protein called NEF basically takes that ADP off and puts another ATP on and opens it up, and then that protein can be released. Okay. Chaperones don't make the protein fold correctly directly. They don't forcibly fold the protein. They just keep a area of the protein protected so it doesn't form aggregates. Okay, and then that protein, when it's protected, can then fold into the favorable state, or it can not. Okay. Right. The other type of chaperone the book mentions are chaperonins. Okay, this is a little bit bigger system. Okay. So in here we have a folding intermediate, okay, delivered by HSP70, and this is in a bacteria cell. And in the bacteria system, this is referred to as the GRO-EL and GRO-ES complex. And what it is there's two pieces to it, two halves. Okay, they're really similar. Okay, the GRO-EL part has 7 chains to it that form this circular structure with a pocket inside, so your folded intermediate can go in there. The GRO-ES is like a top cap that goes onto it and keeps that folded intermediate protein inside. Each one of these has an ATP connected to it on the GRO-EL. Okay, those can be hydrolysis, undergo hydrolysis, hydrolyzed, and by doing that, in the time it takes for that to happen, the protein hopefully has folded into its correct state. Okay, then GRO-ES gets removed, and the native protein comes out, or another folding intermediate comes out, and if that's the case, it can go back into one of these. Okay, and it has two sides, so as one side is doing this, the other side is free, and then when this opens up, a protein can go on the other side before that is the GRO-ES is kicked off the other side. Okay, so there's in this case, there's always one side is always occupied. What this figure doesn't really do a good job of is showing you how the ATP is replenished. Okay, so it's showing you ATP here is being used when that is folded, but it's not showing you how it's recycled. So this isn't a completely accurate or this figure doesn't really offer a complete description of how this works, okay? But the important part to note is that you have the GRO-EL, GRO-ES complex. Your misfolded protein goes in there. There's ATP hydrolysis. Then the ES part the cap comes off. Your protein either comes out in the correct fold or a folding intermediate that can go back in. And when it's in there, it's protected from forming aggregates. This just shows you a more detailed structure of this complex. Okay. Right, the book mentions at the end of this chapter there are many diseases that involve protein misfolding. Okay, one that it really talks about is beta amyloid, is a globular protein. In its native state, it's soluble. It can misfold, partially misfold, and when it does that, well, you'll see here in the native state, there's some beta strand structure and some alpha helical structure to that. In the unfolded state, or partially unfolded, there's the beta structure is separated from the alpha helical structure. And when that happens, two or more of this beta amyloid protein that's misfolded can interact with one another, and they can interact very strongly where that beta structure is, the beta strands. And remember that there's hydrogen bonding that can occur between strands in the beta structure. And so that happens, and that becomes, a very strong interaction. And we get what are known as aggregation, and then ultimately as amyloid fibrils. Okay, so the stacking of these beta strands on one another makes, on the molecular level, we can see that happening, but if this happens enough, you can actually see these on the macroscopic level, and that's what we refer to as fibrils, because they're making fibrous structures within some cells that you can actually see. Okay, and then that fibril structure, it becomes very damaging. Okay. Some other diseases that the book talks about that are protein misfolding diseases. The beta amyloid is part of the problem in things like Parkinson's disease and in Alzheimer's. Tau fibril formation is also important in certain diseases like that. And then it talks about prion diseases. And prion diseases are very similar to where you get a unfolded protein that normally has more of an alpha helical structure. When it unfolds, it can adopt beta conformation, beta sheets. They can stack on top of each other, and that's the basis of prion diseases, things like mad cow disease. Okay. Right, so next time we'll start chapter 5, next lecture, and that's discussing hemoglobin and myoglobin. Okay. And just a reminder that our first in-class activity will be September 22nd, so you want to make sure that you join the class meeting that day. We're going to be talking about how to determine some physical properties of protein just given their sequence, and how to use some of these crystal structure databases and crystal structure viewers.